Hello everyone, my name is Marie and today I will be talking about my PhD research which involves big data in medicine. There are many diseases for which we don't know yet why they occur and what can be done to prevent or treat them. One approach is to analyze many blood samples with powerful computers, and today I will show you how we do this in a research field called bioinformatics. Blood is extremely rich in information since it contains your DNA as well as a lot of other molecules. Some of these molecules are there because of the food you ate, some of them because your body produced them based on your genes, such as ABO. What this means is that your genes can to a certain extent determine which molecules and how many of them will be present in your blood and thus predispose you to develop certain illnesses. Now imagine you have blood samples from many tens of thousands of people who either have a specific disease or not. You could suddenly see which genes and molecules are the root of that disease, but you might also face another problem. Your personal computer is too small to handle all of this data. To avoid that, we as bioinformaticians use so-called supercomputers, which have a capacity to store hundreds of terabytes of data and which have a very high computing power. But there's one last problem. How does the computer know what to do with the data? For that, we write code in programming languages. Maybe you have heard of C, C++, or Python, which contains all the instructions for the computer. Now putting all of this together, we send the code to the supercomputer, which analyzes the data and then sends us back the answers which gene or molecule in our body might be linked to a given disease. And this is exactly what we did for diseases such as asthma, arthritis, and heart diseases. Importantly, once you know what causes the disease, other researchers can then, in the next step, develop medication that target these bad molecules that make you sick. Of course, there are some limitations to our work in that the molecular disease links that we identified are not as reliable as if they came from mice experiments, for instance. But compared to animal experiments, analyzing thousands of human blood samples is much cheaper, faster, and more ethical. And with more data becoming available in the future, we expect that this approach will become more reliable and will benefit the development of medical therapies tremendously.